Welcome everyone to Chapter 6. We'll be discussing the chromosomal and genomic basis of disease. I have outlined here various chromosomal abnormalities and their mechanisms. I'll be discussing them further in detail in this presentation, but I'll just take you through them now just briefly. So abnormal chromosome segregation often results from non-disjunction, causing aneuploidies such as trisomy 21, or monosomy such as 45X, as well as uniparental disomy, or UPD for short. Recurrent and non-recurrent chromosomal rearrangements occur due to genomic hotspots and mechanisms like non-allelic homologous recombination, leading to conditions such as deletions and duplications as well as CNVs, such as the 1p36 deletion syndrome. Unbalanced familial chromosomal abnormalities arise from inherited structural changes and are often observed in offspring of balanced translocations or paracentric inversions. Genomic imprinting abnormalities impact imprinted regions on the chromosome, causing disorders such as Prader-Willi syndrome or Angelman syndrome. And lastly, we'll speak about sex chromosome abnormalities, which add another layer of complexity with diverse phenotypic consequences. Let's begin with aneuploidies involving whole chromosomes, specifically focusing on survivable trisomies such as trisomy 21, 18, and 13. These are viable due to their relatively low gene content because they do involve very small chromosomes, and this reduces the likelihood of lethal developmental defects. All trisomies are associated with congenital abnormalities and intellectual disability. Now, Down syndrome, or trisomy 21, has the highest survivable rate, with the life expectancy extending beyond 50 years. Now, Down syndrome is the leading genetic cause of moderate intellectual disability, and with an incident rate of 1 in 700 live births. Although it's influenced by advanced maternal age, which is over the age of 30, more than 50% of cases are actually born to mothers under the age of 35 years, and this is due to higher birth rates in younger mothers. Now, the characteristic features, which is diagnosed at birth due to distinctive physical traits, are hypotonia, short stature, short neck and loose skin, short, broad hands and single palmar crease, and fifth finger clinodectally. Now, individuals with Down syndrome face a range of clinical challenges, including intellectual disability, as I mentioned, developmental delays, and a heightened risk of congenital heart disease and leukemia. Now, overexpression of genes on chromosome 21 actually contribute to these outcomes. Also, many individuals with Down syndrome exhibit Alzheimer's-like dementia early on in life, indicating the syndrome's broad impact on health. Chromosome analysis is actually really important for confirming a diagnosis of Down syndrome as well as for genetic counseling. Now, approximately 95% of cases result from an extra chromosome 21 due to maternal non-disjunction event, particularly in older mothers. Other cases can involve mosaicism with milder phenotypes or a Robertsonian translocation where chromosome 21 attaches to another chromosome. These cases have different recurrence risk in families. Variants in Down syndrome, such as the rare 21q21q translocation, which is an isochromosome, present unique challenges. So all offspring of an individual with this isochromosome are affected. So they all have Down syndrome. Partial trisomy 21 also involves duplications of a specific region on chromosome 21, and this helps researchers pinpoint areas contributing to Down syndrome traits such as heart defects. Now that we've discussed aneuploidies, let's actually explain what uniparental disomy means. So the definition is when both copies of a chromosome come from the same parent rather than one from each parent. So there's different types. There's isodisomy, so we have two identical sister chromatids, and this is caused by non-segregation in stage 1 meiosis. And there's also heterodisomy, where both homologs from one parent end up in the fetus, and this is caused by non-segregation in stage 2 meiosis. UPD often results from a trisomy rescue, which occurs in a trisomic fetus, so a fetus that carries a trisomic cell line. Trisomy rescue is a corrective mechanism where one of the extra chromosomes in a trisomic zygote is lost during early cell division. This actual process can stabilize the genetic makeup of the fetus, potentially allowing for a viable pregnancy and reducing the likelihood of miscarriage. However, in some cases, it may result in UPD, where both chromosomes of a pair, such as from a chromosome 15 or 21 or 18 or any other chromosome, are actually inherited only from one parent. So what are the clinical implications of UPD? So the associated disorders are seen in conditions involving imprinted regions or recessive diseases. Example would be cystic fibrosis in patients with two maternal copies of chromosome 7, or Prader-Willi syndrome where a patient would have two maternal chromosome 15s. The impact on health can cause disease if there are two copies of imprinted genes from one parent. And it's important for clinicians and genetic counselors to actually find this out to consider in cases from unexplained genetic disorders. Now, structural variants that we'll be discussing from this point onwards, such as inversions and deletions, can lead to a range of genomic disorders. Some are recurrent due to mechanisms such as non-allelic homologous recombination, while others, like Cri-du-Chat syndrome, are non-recurrent. These structural changes are crucial in diagnosing and understanding many genetic diseases. Now, this table is taken straight from your book, and you can have a look at it at a later time, but really the gist of it is that recurrent structural variants are caused by non-allelic homologous recombination and occur at consistent genomic locations in unrelated individuals, and this is due to the presence of segmental duplications. These are often associated with known genomic disorders, whereas non-recurrent structural variants result from mechanisms such as non-homologous end joining, fork stalling and template switching, or microhomology, mediated break-induced replication. These variants exhibit diverse breakpoints and are distributed randomly across the genome, leading to more unique disorders. Now, structural variants that we'll be discussing from this point onwards, such as inversions and deletions, can lead to a range of genomic disorders. Some are recurrent due to mechanisms such as non-allelic homologous recombination, while others, like Cri-du-Chat syndrome, are non-recurrent. These structural changes are crucial in diagnosing and understanding many genetic diseases. Chromosomal inversions are actually structural arrangements where a segment of a chromosome is reversed end to end. In this case of pericentric inversions, the inversion involves the centromere altering both the P and Q arms of the chromosome. These inversions can be visualized through karyotyping, as shown in the example of chromosome 3, where the inversion is through, the pericentric region. While often begins in carriers, they can lead to genetic imbalances during meiosis, potentially increasing the risk of abnormalities in the offspring. Shown here are examples of inversion impact. We have Williams-Beuren syndrome, where there is an inversion on 7q11.23, and this is found in 25% of parent chromosomes of affected probands. 3q29 deletion syndrome involves smaller inversions and found in some parents without causing structural variants. Larger inversions are linked to pathogenic deletions in probands. Structural variants actually arise through different mechanisms. We have non-allelic homologous recombination, which leads to recurrent deletions and duplications, and those have identical breakpoints, while non-homologous end joining causes non-recurrent rearrangements. These two mechanisms underlie many syndromes, especially on chromosome 22, which is actually prone to deletions associated with DiGeorge syndrome as well as some duplication syndromes. The 22q11.2 region is prone to both deletions, leading to DiGeorge syndrome as well as duplications. These rearrangements actually cause distinct syndromes with characteristic features such as craniofacial anomalies, intellectual disabilities, as well as immunodeficiencies in deletions. Duplication syndromes generally have mild effects but still result in congenital abnormalities. As I mentioned, 22q11.2 duplication syndrome is actually a duplication that's generally better tolerated than the deletion syndrome. It's usually caused by reciprocal duplication of the 22q11.2 region and causes unique malformations as well as birth defects. We have Kettai syndrome, which is a tetrasomy of 22q11.2 region, and it is caused by an inversion and a duplication. And the concepts of deletion and duplication syndrome of 22q11.2 actually apply to many other genomic disorders. So the better we understand this region on 22q11.2, which is very much prone to deletions and duplications, the better we would understand how this actually takes place in other regions of other chromosomes and what the consequence is on the carrier. Another recurrent deletion syndrome is Creutzfeldt syndrome, which results from a deletion on chromosome 5p, which is a short arm of chromosome 5, and is named for the characteristic high-pitched cat-like cry in affected infants. This is rare. Actually, this condition is quite rare and it's mostly sporadic, although some cases are due to parental translocations, resulting in an unbalanced form in the fetus. The deletion size often correlates with the severity of intellectual disability and the physical features of the patient. Some deletion syndromes, such as 1p36 deletion syndrome, are non-recurrent, with breakpoints varying between cases. Large deletions can be visible with karyotyping, but the smaller deletions may require advanced techniques, such as microarray analysis if it's suspected, or a targeted technique such as FISH. The mechanism often involves recombination between repetitive elements, leading to that deletion in that region of chromosome 1p. Familial chromosomal abnormalities may be balanced with no immediate effect on the carrier, or unbalanced, leading to dev delays and abnormalities in the fetus. So if we have a known familial translocation, genetic counseling is actually quite important because there is that potential for unbalanced outcome in the offspring. But in regards to de novo balanced translocations, which means non-inherited balanced translocations, they are associated with congenital abnormalities in about 1 in 2,000 live births. And although balanced, there might be a risk of disruption of the genes or gene at the translocation breakpoints, and this is what causes the issue in the carrier. Now we need to focus on the pathogenesis of chromosomal abnormalities due to these structural rearrangements. And we have to highlight how they differ from non-disjunction related abnormalities. Unlike non-disjunction, which causes UPD, aneuploidies such as trisomy 21, monosomy X, these abnormalities actually arise from random chromosomal segregation in the carriers of balanced structural changes. such as translocations or inversions, and this happens during meiosis. Balanced translocations, for example, can form a quadrivalent structure during meiosis, and this increases the risk of producing gametes with unbalanced genetic material, or called derivative chromosomes. This can result in their offspring carrying a genomic imbalance, and also this causes associated clinical consequence in that offspring. A notable example is the inversion of 3 on P25Q21, which is observed in certain Newfoundland families where the risk of abnormalities in offspring actually is greater than 40%. This highlights the importance of understanding structural chromosomal abnormalities for genetic counseling and to assess risk in the carrier and the potential risk to their children. In the next few slides, we'll be discussing neurodevelopmental disorders resulting from unbalanced chromosomal structures. So just an overview of neurodevelopmental disorders. So the definition is any disorder that's affecting cognition, communication, behavior, and motor skills. The prevalence is 2 to 3%. For intellectual disability and developmental delay, and 1% for autism spectrum disorder. The genetic causes are often unclear, and diagnosis aids in management and counseling, but genetic testing doesn't often actually give you an answer for why these disorders arise. Screening could be via karyotyping, microarray, or whole exome genome sequencing. Chromosomal microarray is actually the first-tier diagnostic tool for individuals with intellectual disabilities and autism spectrum disorder, and it has a detection rate of about 12 to 16% of cases. Now, larger de novo, which means non-inherited, chromosomal copy number variants are more commonly associated with intellectual disability than with autism spectrum disorder. An example of syndromic copy number variants include deletions or duplications at regions such as 3q29, 16p11.2, and 22q11.2, which we discussed in earlier slides. Now, hundreds of genes have been implicated in these disorders, with over 1,000 potentially involved in neurodevelopmental conditions. Exome sequencing has a detection rate of about 15% of non-syndromic intellectual disability and autism spectrum disorder, and a lot of doctors actually opt for sequencing if they do get a negative microarray test. Whole genome sequencing also provides a broader scope, identifying both de novo and as well as inherited pathogenic variants, expanding diagnostic capabilities. This is an overview of genomic imprinting disorders. So Prader-Willi syndrome involves the absence of paternally expressed genes on chromosome 15, which means that either both chromosome 15s are inherited only from mum through trisomic rescue, or the paternal region on chromosome 15 is actually deleted. This leads to obesity, intellectual disability, and other features in the carrier. Angelman syndrome, on the other hand, is also on chromosome 15, but it's actually due to the loss of maternally expressed genes, which means that either both chromosome 15s are inherited solely from dad and the maternally expressed genes are absent, or that the maternally expressed genes on chromosome 15 are deleted, leaving only the paternally expressed genes in the carrier. Children often present with severe intellectual disability and a characteristic happy demeanor. They're always smiling. Both syndromes have specific testing methods, which include FISH, and that targets the deleted region on chromosome 15, if there is a suspicion of Prader-Willi or Angelman syndrome, or you can have methylation analysis to see if both chromosomes are methylated in the same way, which means either the paternal or the maternal expressed genes are only inherited. And you also need a trio analysis to see where that the chromosome 15 is originating from. So some of the key features of Prader-Willi syndrome is neonatal hypertonia, which means that there is a decreased muscle tone and the babies actually refuse to eat at the start of life. Later on, you will find that obesity and excessive eating takes place. They also have small hands and feet and very short stature. The genetic cause, as I mentioned, is the absence of paternally expressed genes on chromosome 15. The actual region is 15q11.2 to q13. And over 70% of cases involve a deletion on the paternal chromosome. And this is why FISH is actually quite a useful tool, or even multiplex ligation methylation assays. Testing methods, as I said, is through multiplex ligation assay or a microarray which detects both copy number variation and can also identify UPD. MS-MLPA, which is a methylation MLPA, detects methylation patterns and imprinting errors. And FISH, as I mentioned, targeted for suspected diagnosis of deletions in that region. Unlike Prader-Willi syndrome, Angelman syndrome is actually a little bit harder to determine or detect in a newborn baby. But later on, the key features are severe intellectual disability, ataxia, seizures, and happy demeanor. The only thing as a newborn would be perhaps feeding difficulties, but that is so, non-specific really for a newborn. Genetic cause, as I mentioned, is a loss of maternally expressed genes on chromosome 15. The region is exactly the same. It's a 15q11.2 to q13 as a Prader-Willi syndrome, and is often due to a deletion on the maternal chromosome or a mutation in the UBE3A gene. Testing methods, the same as Prader-Willi. It's multiplex ligation probe amplification as well as microarray, which detects both copy number variations and can help identify UPD. UBE3A sequencing can detect 10% of cases due to point mutations in that region, and FISH is very useful to target for suspected diagnosis of deletions in that region. So sex chromosome abnormalities such as Turner syndrome, which is monosomy X or 45X, as well as Klinefelter syndrome, which is a 47XXY complement, are generally less severe than autosomal aneuploidies. Turner syndrome affects 1 in 4,000 females and often leads to congenital heart defects, infertility, and short stature, while Klinefelter syndrome affects 1 in 650 males and is associated with learning difficulties, androgen deficiency, as well as infertility. So why are sex chromosome aneuploidies less severe than if they involve other chromosomes? Well, this is largely due to the mechanism of X inactivation. In females specifically, one X chromosome is randomly inactivated during early development to balance gene expression with males. However, in cases of structurally abnormal X chromosome or X autosome translocations, the inactivation is often skewed towards the abnormal X to maintain viability. So that means that if you carry an abnormal X in addition to your normal X, that abnormal X is preferably inactivated in your cell. This process involves the X inactivation center, which produces the XIST RNA. This RNA coats the inactive X chromosome, silencing its genes. What is the clinical impact? Well, X-linked intellectual disability is actually significantly more common in males due to their single X chromosome. Currently, approximately 100 genes have been linked to X-linked intellectual disability. Fragile X syndrome is the most prevalent form of X-linked intellectual disability. It's caused by a triplet repeat expansion on the FMR1 gene located on XQ27.3, leading to gene instability as well as silencing. As I mentioned, there is a male prevalence of X-linked intellectual disabilities and disorders, and they are more common in males due to their single X chromosome, and the estimated incidence ranges between 1 in 500 to 1 in 1,000 live births. The most common cause found up to date is the Fragile X syndrome, which is caused by mutations in the FMR1 gene, and this remains the most common identifiable cause in males. Beyond this, there are nearly 100 other X-linked genes which have been implicated largely through family studies. Now, diagnostic advances have been through microarray, which identifies copy number variations, including insertions, deletions in about 10% of cases. Exome sequencing, which highlights excess de novo variants specific to the X chromosome. Long-read sequencing, which effectively detects triplet repeat expansions in the FMR1 gene, and triplet repeat PCR, which enables the detection of both premutation and full mutations on the X chromosome in both females and males. So we've discussed the chromosome X, now we'll move on to chromosome Y. The Y chromosome is actually really gene poor. It contains fewer than 100 genes, and most of these genes are involved in gonadal development and male-specific functions, such as the SRY gene, which is located on the Y. It plays a critical role in initiating male sex determination by triggering the development of testes. There are some factors on the Y chromosome, such as the AZFA, AZFB, and AZFC regions, which are essential for sperm production. So deletions in these regions actually lead to reduced or absent sperm count, and this contributes to male infertility. Sex determination involves both chromosomal configurations, such as the XY for males or XX for females, as well as other genetic factors. Disorders of sex development can arise from mutations or disruptions in these pathways, leading to atypical gonadal or sexual development in the person. So as I mentioned before, aneuploidies involving sex chromosomes are actually less severe than aneuploidies involving other autosomes. This reduced severity is due to mechanisms such as the X inactivation, as well as the low gene content of the Y chromosome. Now, in regards to incidence, Klinefelter syndrome has an incident rate of approximately 1 in 650 males. Triple X syndrome affects 1 in 1,000 females. XYY syndrome affects 1 in 1,000 males. And Turner syndrome is actually the most common abnormality found in spontaneous abortions and found in 1 in 4,000 live births. The clinical impact of Klinefelter syndrome is associated with learning and language difficulties, with an increased risk of depression, androgen deficiency, and infertility. And Turner syndrome presents with an increased risk of congenital heart defect, infertility, and they're usually short stature. Interesting facts though, triple X females are usually really tall, and XYY males are usually, well, there was a study done a few years ago, and they found that a lot of XYY males end up in prison due to the high testosterone. Yeah, there you go. Androgen insensitivity syndrome is an X-linked condition which affects 46 XY individuals who lack functional androgen receptors. Complete androgen insensitivity presents with female external genitalia and sparse body hair despite a 47XY complement. Diagnosis actually relies on androgen receptor gene sequencing and hormone testing to confirm androgen insensitivity. Those patients actually are detected by accident through a karyotype. They present as female. They come for a karyotype due to some sort of intellectual disability or other infertility issues, and we find that there is a 46XY complement on the chromosomes. And our first thought is, oh, have we mixed the samples? Why does this patient have a male complement when it's clearly a female? And then we do further testing, and this is when we find that there is a androgen um, sensitivity issue. So there's an AR gene mutation that results in a, a dysfunctional androgen receptor and causes this problem in the carrier. So some of the diagnostic methods for androgen insensitivity would be through AR gene sequencing, and the purpose is to identify mutations in the AR gene, and it really confirms an AIS diagnosis and distinguishes between complete and partial forms. We can also do hormone testing, and the purpose of this is to measure the androgen levels, so testosterone and dihydrotestosterone, and to confirm the normal androgen production despite lack of response. We can also do a pelvic ultrasound, and this visualizes internal reproductive structures and detects absence of uterus and the fallopian tubes and confirms the presence of undescended testes. Also karyotyping, and this is where a lot of the cases are actually detected, and the purpose of this is to determine genetic sex. So a lot of the time, as I said, they come in as females, they come in for infertility or other um, issues, and we detect a 46XY complement and do further testing. So it either starts with a genetic test and flows down to the confirmatory karyotype, or it starts with the karyotype and moves up to confirmation via genetic testing and via sequencing. So finally, let's discuss diagnostic technologies such as microarrays, short-read sequencing, and long-read sequencing, which provide a comprehensive toolkit for detecting structural and copy number variants. So microarrays are really the first tier testing. This is what a lot of GPs ask for to start with because it's cost effective, it's fast, and it's commonly used for detecting copy number variants or SNVs. Short-read sequencing includes whole genome and whole exome and primarily targets single nucleotide variations or SNVs. Long-read sequencing enhances the detection of complex structural variants or SVs, which are often missed by short-read sequencing, and also is very useful for things like Fragile X syndrome and triplet repeat disorders. We do rely on advanced structural variant detection, such as algorithms and toolkits, and also we look at population frequencies. We look at NOMAD, which help assess the frequency of that particular variant in the normal population. So if we find that it is present in the normal population at a high rate, that means it's highly non-pathogenic. But if it's not present in the normal population, that means it's likely or could be pathogenic. Additional techniques that we do look at, such as optical mapping, enhance the resolution for complex structural variants and is crucial for diagnosing deletion and duplication syndrome. It really excels in resolving those complex structural variants, and um, it's particularly useful in diagnosing deletion and duplication syndromes.